clinical trial space, I think we're all familiar with the challenge of obtaining imaging data, processing imaging data, quantifying them, the amount of intense human effort that takes. There hasn't been an encounter with Sarah that imaging hasn't come up. The challenges and opportunities in that context, and it's so existential to the care and research we do. I think this is one of the areas that CBTN is going to lean in extremely heavily on. One piece of the ARPA-H effort is focused on imaging with our partner Flywheel, and you'll see a preview of that work with Ariana.
So I originally started making this slide and I was attempting to put logos of organizations and bodies that have been contributing to and putting active, amazing effort into data generation efforts, many of which we've seen over the past 24 hours or more. However, out of fear of accidentally forgetting anyone, I just replaced it with this image. So there's a ton of amazing data being generated at many different levels and spatial scales. There's lots of diverse data modalities. Amazing data sets like the CBTN data set that are often delivered on these flexible analytical platforms. That in the pediatric brain tumor space is really leading to the generation of these predictive models. So again, as we've seen and continue to see this afternoon, there are novel tumor characterizations coming out of methylation, immune-based clustering, combined with radiology features that are developing these new ways of thinking about how do we consider patients relative to other patients, what are the types of genetic pathways that underlie and the biological mechanisms. And there's no doubt that this is going to pave the way for novel therapeutics in pediatrics. But thinking about the technical infrastructure that underlays a lot of these development efforts, if this is kind of at a high level what a research workflow might look like for a given experiment. If a researcher is lucky, there's a data set that's been generated for them. They obviously might generate a data set as part of their lab or their wider team. But they'll access those data files, they'll generate them at their institution, let's say. They'll port them into, most often, a modality-specific software platform. This could be a local software. Oftentimes, people download it to their computer. They'll run data preparation. They might also use one of these platforms like Cavatica for genomic data processing or Flywheel for imaging. And they'll create derived data output. So they'll prepare that data, they'll extract hopefully relevant features for whatever prediction they're trying to make, and they'll run those statistical or AI ML algorithms in a lab, oftentimes maybe an institutional cluster. If they're lucky, they'll get to use a cloud-based computing environment, and they'll build out, let's say that they're, with a little bit of luck and a lot of iteration, they're able to build a well-performing predictive model for the data set that they're working with. Then they go to publish this essentially prototype of an AI ML model or statistical model, and oftentimes they'll release it on a platform such as GitHub, which is essentially just a code repository. So they'll deposit their software code in a place essentially where other people can go on the web browser, make it public. They'll often put the URL in their publication, and essentially this is where the model stops. I'm not saying that's true for everyone. Please don't get offended if you're, continuing and releasing your model, but this is, for a lot of cases, here I'm just showing you for imaging specific models that have been developed for brain tumors, this is essentially the end all be all for our models. And just to kind of highlight, these don't get updated. However, the software that they depend on often does get updated. And so a year later, it becomes sometimes impossible to recreate and run that model on. Let's say I'm a researcher, I have a new patient data files that I want to run through the model. I can't do that. Recently, there's been some amazing web hosting that's been done. Again, this creates a user-friendly environment. It's great for running, especially for single cases. However, not scalable. Oftentimes, they only take a single sample at a time. It's not interoperable with our data portal platforms. And so really there needs to be a fill of this gap between the model design prototyping stage and the testing of that on a single data set to the deployment production ready AI ML models. So there really needs to be kind of a push from I've created a model on a data set, but now does that model generalize to data from other institutions, for example? Can I test that model over years on new data coming in? How does it perform? Can I update the model? Can I account for data set differences and biases if there's differences in populations that the data is being generated from? So as an effort towards kind of this, what's called an ML ops framework, machine learning operations, we started with a use case of our imaging AI-based models that are relatively new. So if you don't already know, radiology imaging is central to assessment of tumor burden at a given time point, but particularly over time. So it's one of the non-invasive methods of being able to track the status of a tumor, again, as a patient, for example, is going through different treatment options, potentially if they don't receive surgery, if it's a certain type of tumor that's non-resectable, imaging will be used. And so they go into the clinic, they receive MRI exams at regular intervals. Let's see, do I have a cursor? No, that's okay. Or a laser? No, that's okay. It's okay. So standard of care essentially is what I'm showing you here. It's a manual 2D measurement of essentially what the goal is the maximal diameter to capture the largest size of the tumor at a given time point. Recently, there's been some promising AI models, deep learning based, that can actually generate 3D volumetric. Thank you. Sweet. There we go. That can generate these 3D volumes. So the idea here being that 2D measurements often rely on a single radiologist. There's a ton of difficulty sometimes in cases where consensus is needed and might not be necessarily possible. So the idea here being that AI potentially can produce more consistent measurements across time and kind of help the radiologists and the clinical care team make these decisions about whether a tumor, for example, is growing, shrinking, et cetera. So in the pediatric brain tumor space, we have a couple of promising models. And again, the goal here was to take those models, provide them to the CVTN and the broader community in a cloud-based environment. So a user can essentially click through, they can run these models on their imaging data set, whether it's a single file or an entire data set. And also we can create new ground truth data sets for continuous benchmarking and run those back through the models to continue to update them and run model updating and continuous monitoring. So again, an eagle level view of the overarching workflow. Typically what's done is the images will be selected. They'll be run through standard pre-processing to kind of account for some of the noise generated at the scanner level. They'll be pre-processed and then run through these deep learning models, at which point, because these models are not perfect yet, they're reviewed by a person who has relevant expertise, such as a radiologist, and they're manually refined, they're updated, cleaned up, and then the idea here being that creates new ground truth that can be further and iteratively put back into the model for further tuning and updating. We also can take those reviewed and quality controlled segmentations and create measurements of the tumor at a given time and over time. All right, so Flywheel is our basis here because that is the platform that we deliver the radiology and digital pathology, but of course, in this case, the MRIs to our CBTN investigators and researchers at no cost. So researchers will request access, they'll get a user account and they log in and they'll see the radiology MRIs and Flywheel. So that was our starting point for building out a ML ops workflow for tumor segmentation. Over the past several years, we've built in standard pre-processing pipelines. That's not necessarily, oh no, 5 minutes, okay. All right, we'll skip past that. So we built in pre-processing tools. Those are readily available. Okay, sweet. And we also built in, thanks to an amazing solutions architect at AWS who built out this prototype, JP Leger, we ported our models into AWS SageMaker. And I'm going to talk about that a little bit more later, but essentially the idea here is that we have created a central kind of ground source of truth for our deep learning models, and Flywheel can source those and essentially run them on demand. We also have now the ability to run that manual tumor refinement in Flywheel. So I'm going to show you what that looks like. But again, all of this now is integrated between Flywheel and AWS so that there's a kind of single source of file management and a single source of model management. Right. So now I'm going to show you what that looks like from the user's point of view. So again, just to orient you, this is the landing page of Flywheel. A user would come in, they're able to find those pre-processing tools, the data image preparation tools. Here I've selected the winning tumor segmentation model from the BRATS Community Challenge for pediatric tumor segmentation, and that's from a team at Children's National led by Dr. Marius Linguraru. Here I'm inputting for a single time point the pre-processed images into that gear. And so now it's running on AWS. I'm going to then essentially I've cheated here. I've put a predicted model prediction file in there already. And I went to a drop down menu and I'm opening now a session in AWS AppStream that is opening up a separate software called ITK Snap, which has fancy tumor segmentation editing tools. So here it's opened up those images from that given exam. I'm opening up the model predicted tumor segmentation. And so we can see it there in the colored overlay. I can scroll through. There's relatively no latency. So even though this is running in a web browser and not on your local computer, it responds very quickly to my clicks. Here I'm just showing you what that model predicted segmentation looks like. I can flip through different types of images. And let's say I wanted to go make an edit here. Of course, this is just for demonstration. So I just pick to color red over green. And I make a click. So I've edited it. And now I'm just saving my edited segmentation. And what that does is that saves that immediately back to Flywheel and integrated with S3. So that then becomes a now file that can be utilized in further analytics. The measurement of that tumor volume can be extracted. It becomes a searchable entity in Flywheel. Here I've used a Jupyter Notebook in Flywheel to plot the tumor volume over time. So again, this particular patient has multiple time points. And so with these now manually edited and QC segmentations, we can look at the tumor over time and compare also to the data set of similar subjects. Now that ground truth file becomes integrated with our central source of truth file management system on AWS, which Flywheel points to and references. That also can now be released on different downstream platforms. And the final point is that because we've deployed these models in SageMaker, they can both be sourced by Flywheel, but they can also be accessible on other platforms as well. So you can create, for example, website if you wanted to deploy your model there. And any now updates to that model can get propagated to wherever those downstream portals are. The model repository in that way acts as a source of truth. We can run model updating with fancy user dashboards. We can essentially perform continuous integration and continuous deployment to really continue to grow the predictive capacity of the model, continue to update it, and continue to release it to the wider community. And this is both true for AI/ML models, statistical models, and also feature sets that are extracted to eliminate the need for researchers to continue to generate these for the same data set. So I hope that I've convinced you that the community has been amazing in terms of building out fair data and fair data sets, and those are a reality, but I urge you to think about fair models and fair analytics. Really, this is just an example, but the idea here is that we can have these amazing data generation and data access portals, but we can also have the same for our end users and consumers for analytics. And really, this is the only way we're going to move towards patient level, patient-centric analysis and comprehensive visualization with these amazing AI/ML models that are coming out of the research, that some of which that we've seen here today. So with that, this was a proof of concept solution. Really here, the goal is to enable the translation of these models back to clinical care. And the only way to do that is going to be to make sure that they perform across different institutions, across different populations. And this is one solution to be able to support that. Thank you, everyone. Again, especially JP at AWS. Everyone here was instrumental to the success of this and is going to continue to work towards this. Thank you.
I don't have a question so much as a comment. I think I heard you mention yesterday that you have how many images?
So there's like over 1,800 patients.
1,800
over 1,800 CBTN patients, and that's amassed over like 1.2 million files.
Yeah, just 1.2 million files. So I just wanted to recognize that, right? Because that's an amazing amount of really tedious work that is really benefiting everyone, right? And being able to build out these models in this way hasn't been done before, and it's going to be something that helps not just within CBTN, right? But other people are going to use this model and move forward. And I don't know, I think like seeing all that is amazing and wonderful, but it kind of skims over the fact that somebody is sitting there making this all happen. And I just wanted to thank you because I know a little bit about the amount of work that went into that. And we really appreciate it.
Thank you so much. I appreciate it.
Can I ask a question this is Warren on Chime. Can you hear me?
Yes. Hi, Warren.
Hey, that was a wonderful presentation. And I know you didn't have very much time to go through an awful lot of work, but I think one of the components that you didn't get a chance to talk about was the ability to run different versions of a model and compare their outputs. And I think that's so fundamental to building again, I guess I'll say a trustworthy framework for using AI and actually just using computation in general. that it's worth highlighting that I believe that's something that that you've built into your whole architecture.
Yes, definitely. Yeah, thank you. So that's Dr. Kibbe, Deputy Director of Data Science at NCI. Yes, got that right. Yes, that's exactly right. So the idea here is we've started essentially with version 0 or 1, whatever you want to call it, of these 2 tumor segmentation models. Let's say that those get updated. We can both look on Flywheel for different versioning of those models, what the outputs look like, how they relate to those ground truth benchmarking data sets. We can also look at that in AWS if so desired to, again, look at more sophisticated methods in SageMaker of accounting for things like what that testing data set looks like, those features, those models, they're all versioned in SageMaker in that way.
I have one last thing for you, Ariana. First of all, I love the slide of like all the tools on GitHub, because I think anyone who's done any analysis in any field often feels that way. But I also think, the more we do this, the more it's recognized, like, people are always going to build great models. People are always going to great tools. Like, the hard part is what you just showed of that process between the, computer model and then the human manual input. And as a, kind of more on the genomics side, we have this privilege where we think that we can just run all these tools and we don't have to have this manual input. On the imaging side, it's really, I've really learned it's really an existential thing that people have to look at it, they have to tweak it. And I think the rest of the data modalities can really learn from what you have done. I think even on the genomic side, we don't look at the variants until we're at the very end. And then it's like, what happened? Why is there so many? Do I know what's going on? And so I think there is a lot of potential, not just from the imaging standpoint, but across that or across even some of the clinical data. I think Tamara Miller is here, and we'll be talking about Extract EHR and how the clinical knowledge of curating and getting the EHR data and the clinical data, you can even think of similar workflows of being able to do the automated models, curate it manually, and have that feedback in as opposed to just being, the endpoint. So I guess also more of a comment, but I also think that it's very incredible how imaging is pushing on that.
Yeah, thank you. I think that's a great point. It's not that these manual steps are necessarily going to go away anytime soon. So it's about really creating an environment where it's easy to do those types of things and it's all traceable.